Dear student, today we will be discussing mutation. Now, what is mutation? Mutation is a heritable change in DNA of an organism that occurred spontaneously or it is randomly induced. This change is called mutation. Now, let's study the mutation under the heading variation. Variation is of two types, heritable and non-heritable. Non-heritable is influenced by mainly environmental factor. Heritable mutations are of two types, continuous and discontinuous. Continuous, which occurs due to crossing over during the meiosis. Discontinuous mutations may be spontaneous or induced. Spontaneous are natural mutation which occurs at slow rate, and induced are the one which are artificially induced by artificial agent like chemical agent or physical agent, and they are fast mutation, means occur at high rate. Okay, now let's study the mutation under three different headings: gene mutation, chromosomal mutation and chromosomal aberrations. Now gene mutation is of, you can say, four different categories: point mutation, frame shift mutation, transition and transversion. Point mutation again is of different types: nonsense mutation, missense mutation, same sense mutation. Frame shift mutation occurs by addition and deletion. Transition mutation, where the purine is replaced by purine and pyrimidine is replaced by pyrimidine. You know that purines and pyrimidines are the nitrogenous bases which are present in the DNA, and adenine and guanine are called purine, whereas cytosine and thymine are called pyrimidine. So transversion, where purine is replaced by pyrimidine or pyrimidine is replaced by purine. Now chromosomal mutation are due to the change in the number of chromosome. This is also called as ploidy. Chromosomal aberration as the change in the structure of chromosome. This occurs by deletion, inversion, duplication and translocation. Now let's see what is ploidy. So change in the number of chromosome. It is of two types, euploidy and aneuploidy. Euploidy, addition or deletion of one complete set of chromosome. Aneuploidy, addition or deletion of one or more number of chromosome. So euploidy, haploidy or polyploidy. Haploidy is also known as monoploidy, where there is loss of complete one set of chromosome. For example, the organism is diploid, so if one set of chromosome is lost, the organism will become haploid. Polyploidy means addition of one full set of chromosome. For example, the organism is diploid, so one set is added, it becomes triploid, tetraploid and so on. Aneuploidy arises due to failure of separation of homologous chromosome during meiosis. This process is also known as disjunction. Now it is of several types. Monosomy. In the monosomy, there is loss of one chromosome, one sex chromosome. For example, Turner syndrome. So this is called as monosomy of sex chromosome, where one X chromosome is missing. Okay. Nullisomy, where two of the sex chromosome are missing. So this is called nullisomy. Polysomy is again where three pairs of chromosomes are there added. Okay. So this effect is, polysomic effect is by the addition of one or more chromosome. See, when the disjunction occur at meiosis, then suppose one chromosome is going to one gamete, then definitely the another gamete which is simultaneously forming will have one copy of that chromosome less. Okay, So it depends that in the gamete what is expressed, according to that it is known as that particular somy. Now if you see trisomy, three different examples are given over here. Trisomy, tetrasomy, or what we have in our syllabus, trisomy. So for example, Klinefelter syndrome. Okay, this is called as trisomy of sex chromosome, in which one extra sex chromosome is present in the male. Down syndrome is called trisomy of 21st you can say chromosome. So this is related to autosomes, okay, in which 21st chromosome is you can say extra. And Patau syndrome, this is called as trisomy of 13th chromosome, in which the autosome 13th number chromosome is extra in an organism. So this is how we study the ploidies. Now let's come to the chromosomal mutation or aberrations. So let's consider over here. See this sequence of gene on the chromosome. Sorry, just a minute. First we'll just go through what are mutagens. Okay, so mutagens are the agent that induces mutation, could be chemical or physical. Chemical mutagens are ethyl methane sulfonate, sodium azide, ecridine. Okay, and physical mutagens X-rays, gamma rays, UV rays, etc. Now mutations are not always harmful. In next few chapter we are going to see how the mutation is used as tool in the genetic engineering experiments. Now what is mutant? It is the smallest portion of gene that can mutate or change. It may be single nitrogenous base. So a gene has many mutants. Okay, it depends how the mutation could be of 2 base pair, 3 base pair, of the whole gene, of the whole chromosome. Now let's understand this. Now 1 to 14 over here, I've given number to the nitrogenous bases. So this represent, let's say this represent the sequence of nitrogenous base in DNA, or we can say this is a part of DNA, or we can say this is a gene. Okay, so it has a specific sequence where 14 nucleotides are present. Now to understand the mutation, let's focus on 9, 10 and 11 position. So if I read the frame, it is GTT at 9, 10 and 11, and at 8 it is A. So 8, 9, 10, let's focus on this. 8 is A, G, T. Now suppose there is deletion of G from position 9. Okay, so if there is deletion of G from position 9, then whatever is position 10, that is T, will be shifted at 9, the T at 11 will be shifted at 10. So here we can say GTT is replaced by ATT after deletion of the G from 9 position. If you look at the addition of extra base between 9 and 10, actually it is at 10th position. Okay, so at 9 we have G, at 10th there is extra base which is added, which is A. So rest of the shifting takes place toward the right side. Then T will be at 11, another T will be at 12. Okay, so you can see GTT over here is replaced by GAT. Okay, just by addition of one base pair. So gene mutation occurring due to change in a single nucleotide called point mutation. Both deletion and addition of nucleotide shift the reading frame of codon from the site of change onward. Therefore is also known as frame shift mutation. Since here shifting of frame takes place because of addition and deletion, so we say this is a frame shift mutation. Now let's see what is substitution. Is of two occur by two different type, transition and transversion. So usually the base pairing is ATGC, but suppose the mutation occurs at position where there is pyrimidine, then pyrimidine is replaced by suppose purine, then AT base pair is changes by GC base pairing. Suppose over here A is replaced by G, so C is going to make complementary base pairing with it. Okay, so this is how it changes. Transversion, purine replaced by pyrimidine. Okay, so suppose in the DNA sequence you have cytosine, and suppose cytosine is replaced by adenine. So complementary base pairing for adenine will be thymine. So here purine will be replaced by pyrimidine. Now sense mutation, again you consider a sequence of gene over here, AAA, GCA, AGT. So what happens in the nonsense mutation? Let's consider position 6, 7 and 8. So 6, 7 and 8 AGT. Now suppose at 7 position G is replaced by T. Okay, so over here the ATT will have, okay, so what will happen? 6, 7 and 8. Okay, so AGT is there. If G is replaced by T, then it will become ATT. So ATT will have UAA on mRNA. Now there is a process called transcription in which the DNA form the complementary mRNA. Okay, only the difference is that when it forms the mRNA, the thymine is replaced by uracil. So ATT will have UAA on mRNA, which is termination codon. There are different types of codon. We will come to this in next chapter. And wherever this termination codon come, the protein synthesis stops there. As the mutation produces termination codon, so coding message is interrupted. Okay. Now look at 1, 2 and 3 position. 1, 2 and 3 is AAA, where A over here is replaced by T. So AAA is replaced by ATA. So AAA will have, actually when AAA was present, then its mRNA was UUU, which codes for the amino acid phenylalanine. Whereas for ATA, mRNA will be UAU, which codes for tyrosine, is another, you can say, amino acid. So modified non-functional product is formed. So wherever in the protein there has to be phenylalanine, instead of that tyrosine is present and defective protein is produced. Such kind of case we have already studied in sickle cell anemia, where defective product is formed, so the RBC shape changes to sickle shape. Now look at 4, 5, 6 position. It is GCA. Now over here A is replaced by T. So when A is replaced by T, actually when it was 4, 5 and 6. So AGT and GCT both codes for the same amino acid arginine. So if you see their complementary RNA, they will be forming AGT and GCT, both codes for the same amino acid arginine. So whatever, there are more than 2 codon which codes for the same amino acids. Okay, so change in codon codes for the same amino acid, hence there is no change in the final product. It will remain the same. So although the mutation occurred, but there is no change in the protein that is found. So this is called same sense mutation. Now look at the chromosomal aberration. Now this represents the chromosome where A, B, C, D, E represent the sequence of gene. So let's see how the mutation occur in the part of chromosome. So deletion, loss of one complete gene. Suppose A, B, C, D, E, F are there. C is lost from here. Okay, so this is called chromosomal mutation by deletion. Similarly, if C is duplicated, A, B, C, C, D, E, F. So one copy of extra particular gene is present. This is called duplication. Inversion, a portion of chromosome break off, turn around and join again in such a way that sequence of gene get reverted. So A, B, C. So from here the breakage occurs and then inverted position it is joined. So E, D, C, this sequence is changed. Now translocation, a section of chromosome breaks and join another non-homologous chromosome. So from some other chromosome X, Y, Z is added. Okay, so this is called chromosomal mutation. So this is all about mutation. Next part we will continue in part 2. Thank you.